welcome back to our channel Biolab, and this is Vamsi here at Biolab, and this is our first live of our channel, guys. So likes should be very hard. You have to hit likes very hard. Come on, please like the video. We'll be continuing with this chapter, that is excretory products and elimination. I'll be trying to complete this chapter max in this lecture, or else we'll be making a small lecture. I'll be making it a small lecture. So yes, please like it. Please like the video. We'll be starting within a few seconds, guys, till everybody joins. That 2 watching should become 10 watching. Then I'll be starting very fast. 10 or 5 watching. Then I'll be starting that. There's 3 watching. There are 3 members watching. There is only 1 like. Please hit that like button, guys. Come on. Still 2 more watching. If we get still 2 more watching, then we'll be starting, guys. We'll be starting then. Come on. Hit the like button fast. So guys, please come on, please increase the watching and also likes. I'll be starting the lecture. Come on. It is getting delayed, guys. It is getting delayed. As much as it gets delayed, we will be lacking in our syllabus, guys. So the thing which we have done is the timetable would get worse. It's going to wane. Come on, come on, come on. Come on, guys, it is the first live of our channel. You are even not liking it. How come this is possible? This is the first lecture live of our channel. Okay, it has been some 6 to 5 minutes since I have started the live. The time is getting wasted. And if anyone has joined still yet, they will be joining in the later lecture, I hope. And if anyone is not watching the lecture, they might watch the recorded part. So yes, let us start, guys. The time is getting wasted now. Let us start. If you have any problem, please mention in the live chat. Okay, if you have any problem, please mention in the live chat. I'm starting the lecture, and today's lecture we will be discussing about the mechanism of concentration of urine, guys. Before going into this topic, I have a small thing to discuss with you, that it is the types of nephrons. Types of nephrons. I have to discuss this topic with you. I haven't discussed this. Types of nephrons. There are basically two types, guys, in nephrons. There are basically two types of nephrons, which are mentioned in our NCERT based upon their location. Based upon their location. See, when you closely examine the structure of the nephron, which we have done long back, so wait a minute, let me try to draw the structure. Okay, we have seen something like this. PCT part, Henry Stroop, collecting duct, DCT part, and all. So wait a minute, guys, please wait a minute. I'll be just back. So yes, here, come back here again. This is your Malpighian body or Bowman's capsule and glomerulus. This is your PCT part. This is your descending limb. This is your hairpin bend. This is your DCT part. This is your CD part. Now the thing is, there are two types of nephrons basically. Here what happens is, in the nephron, this part, see, until the PCT part, here these lines which I am representing here, let me try to use something, these galaxy pen lines, okay, this galaxy multiple color pen lines which I have drawn, the dotted line which I have drawn is a partition. This partition indicates, it separates the medulla from cortex, guys. I have already taught you in the first lecture that kidney is having two parts, medullary part and cortex part. So this line separates both. These kidneys are present in both cortex and the medulla part. Even these kidneys are present in both cortex and the medullary part. Now the part which is above this line, above this Milky Way pen, it is called as cortex. This part, the cortex part, consists of the Bowman's capsule, glomerulus, PCT, DCT part, and the initial collecting duct, ICD part. These parts come under or include under the cortex part. Now the parts which are below this line, say for suppose medulla part, they count. Say for suppose this DL, hairpin bend, and this is your AL, ascending limb, thin and thick, both. These all are constituted, or they are present in the medullary part of the kidney. So this is the thing which you have to remember, and based on this there are two types of nephrons. One is cortical nephrons and juxtamedullary nephrons. Okay, let me write it with some other pen. Cortical nephrons and juxtamedullary nephrons. These are the two types of nephrons. What are cortical and what are juxtamedullary nephrons? These cortical nephrons are the nephrons whose hairpin bend is very small. It is very small. Their vasa recta is either reduced. It is either reduced or absent. These cortical nephrons, in these cortical nephrons, vasa recta, it is either reduced or it is absent. Now this part, see here, if this is what if this is a cortical nephron? Here I have drawn, drew a structure of nephron. If this becomes a cortical nephron, what happens to this? Means the cortical nephron is a nephron whose most of the part is present in the cortex. So that is why what happens is, hairpin band is small. Henle's loop is small. Most of the part it is present in the cortex part. So the Henry's loop, if it is suppose here, the Henle's loop is small. These all points I am not mentioning on the board. Please write down in your notes. I have already mentioned the story of in the Instagram that please get your notes as well as your NCERT textbooks. So write that in cortical nephrons, the hairpin bend or loop of Henle is reduced in the size. You can simply write it is smaller than the juxtamedullary nephrons. In juxtamedullary nephrons, as the name suggests, most of the part it is present in the medulla, and therefore this hairpin bend extends. So see, these are your juxtamedullary nephrons. So these are the two types of nephrons which are present in our kidney as per our NCERT. Okay, now why did I say this? This I have said because it is given in your NCERT. First thing, I haven't explained to you in the previous lectures. The second thing, and the third thing is I have to tell you the parts. In order to continue today's lecture, I have to tell you the parts which are included in the cortex part and the medulla part. So for that I have also covered this topic. And now we will be moving on to today's lecture, the awaited lecture, that is your mechanism of concentration of urine. How this urine concentration, how this concentration of urine is varying with the medullary fluid. So that is what we will be seeing now. Okay, now let's start with the mechanism concentration of urine. So guys, this topic is very simple. This is a very simple topic that we are learning here. So if you just put your brain or use your brain for the application part, it is going to be very simple. Now the thing is, guys, I have told you about the reabsorption already. In this reabsorption, what I have told, the primary urine which is produced is more diluted. It is consisting of much of water. If you excrete that urine, that primary urine as a waste, we'll be getting dehydrated. So the thing is, what our mechanism has adapted is, it can regulate the ionic balance, it can regulate the increase in the pressure, and all those is happening. But why we require this reabsorption is in order to save the nutrients, the essential nutrients like glucose, amino acids. We have different sorts, NaCl, we have urea. In order to save these all from excreting, we have the mechanism of reabsorption. So now see here, this is your Bowman's capsule part. This is your Bowman's capsule part. Okay, let me choose a different color. This is completely matched. Okay, this is your Bowman's capsule part. This part is your Bowman's capsule part. Now the primary urine from the Bowman's capsule enters the PCT carefully, but from the glomerulus, the primary urine enters into the Bowman's capsule, and this primary urine is more diluted, consisting of much of water. So it is having much of water. Next, in the PCT part, I have told you that 70 to 80% of the absorption, 70 to 80% of absorption takes place in PCT. Now water is being reabsorbed. Being reabsorbed. This is your flow of urine, guys. Please remember the flow of urine, not blood. This vasa recta is concerned with the blood. Please do not keep this vasa recta in your mind now. Remove this vasa recta from here. Remove this vasa recta from your mind. We will be coming to that after a little while. Now this urine, while it is moving down the descending limb, the water is getting reabsorbed. Descending limb is permeable to water, impermeable to ions, which means concentration is increasing here. So see, it was initially some 300, which is approximately equal to the concentration of the outer medullary fluid. So initially it was 300 in the PCT part, and while it was moving down in the DL, descending limb, it was 400, and the concentration of the outer part or the medullary fluid is 600. It is okay. And now please observe carefully. From 400 it has increased to 600. From 600 it has increased to 800. From 800 its concentration 1000 to 1200. Now what are you observing here? The concentration from 300 has increased to 1200. The concentration here, it was initially in the initial parts, this is your PCT part, this is your DL part. While moving down, the concentration of the urine which is present in the DL part was increasing gradually, and when it came down to the hairpin bend, it became almost equal to the outer medullary part. So it was initially it was 300 here, it was 400 here, somewhere here, it was 600 somewhere here, it was 800, it was 1000, it was 1200. The outside medullary fluid part is also having 1200 of pressure. Now what you say is, while coming down, the concentration of the urine which is present inside the DL part increases gradually, and dilution decreases, concentration increases, and at a particular point of hairpin bend, on reaching the hairpin bend, the concentration of this urine would be equal to the concentration of outer medullary fluid. So that is why we say it is in what? It is in isotonic medium. It is acting like an isotonic medium. Now, while you are moving up the ascending limb, it is permeable to ions, impermeable to water, which means concentration decreases. See here, initially it was 1200 starting, it became some 800. From 800 it became 600, from 600 400, 400 to 300. Now the concentration decreased gradually. means dilution increased, dilution is increased, and when coming here the concentration has been much increased, and we humans and we mammals produce much concentrated urine, we produce hypertonic urine, we produce concentrated urine, we produce concentrated urine, mammals produce concentrated urine. Now what is this mechanism of concentration? This all I have completed in the previous lecture. Now what is this? Let me take you through our NCERT so that it would be very clear to you all. Let me open the NCERT, 1 minute. Just wait a minute for me. I am opening your NCERT. I am opening the NCERT here. And we will be going through this line by line from our NCERT. Okay. One minute guys, I am just opening. So yes, hope NCERT is visible to you all. NCERT is visible to you all, I hope. Now, what's your NCERT? We will be going line by line in our NCERT itself. From NCERT we will be learning. So, start here. The mechanism of concentration of the filtrate or urine, you can say. Mammals, we have the ability to produce concentrated urine. Now only I explained the way we produce concentrated urine. Since we reabsorb most of the materials and we only release some harmful materials by secreting them back into the urine. And the Henle's loop and vasa recta play a significant role in providing or in producing concentrated urine, and also in the regulation of mechanism of concentration of the filtrate. How these two play a major role, he is explaining here. See this. This flow of filtrate in the two limbs of Henle's loop is in opposite directions. This is, of course, this is obvious. See here, this is your descending limb. The flow of urine here, it is downwards and this is upwards. This flow of urine in the opposite directions in two limbs, we just call it as like this. See, some is like this is your descending limb, this is your ascending limb. So the cap of this pen is here, the cap of this pen is here. So what do you call it? They are in counter current direction, and this counter current direction in your Henle's loop helps in maintaining the concentration or in the regulating the concentration of the urine produced. And also, see here, and also the flow of blood through the two limbs of vasa recta is also counter current pattern. If this is your vasa recta, guys. Now in vasa recta, the blood comes from peritubular network. It flows down like this. It goes to renal venules and renal veins. So that is why this becomes your descending limb. This becomes your ascending limb. Descending limb, ascending limb. Now in descending limb, urine is coming down. Sorry, the blood is coming down, and in the ascending limb, the blood is flowing up. So this also follows the counter current flow pattern. This also helps in maintaining regulating the concentration or regulating the increased osmolarity towards the inner medullary interstitium. Now the medullary interstitium or the medullary fluid, it is increasing. The osmolarity or the concentration of the medullary fluid is increasing from 300 gradually to 1200, 300, 600, 900, 1200. Now this increasing thing, there is some message. Dr. Rohit Roy, अभी मनीष सर का क्लास करे के अय्या. Okay. Now, the concentration is gradually increasing from 300 to 1200. From 300 to 1200, the concentration of the medullary fluid. But the concentration of the urine which is flowing through the nephron, is also gradually increasing and gradually decreasing. So this increasing osmolarity should be maintained properly, and this is maintained by the points which we have discussed just now. What are those points which we have discussed? The counter current flow between the two limbs, one the descending limb and the ascending limb of the loop of Henle, and also between the descending limb and ascending limb of vasa recta. These maintain the counter current flow, and through this the osmolarity is being maintained. And there is also one more point which is helping in maintaining the increased osmolarity, that is your proximity between Henle's loop and vasa recta. Proximity. These Henle's loop and vasa recta are just side by side. So in your NCERT, if you go to this diagram in the structure of nephron, okay, this is your information. See, here they are in close proximity to each other. This red color part, you see this red color part, this is your vasa recta, and this yellow color part, it is your nephron part. And these two are in close proximity to each other. These two are in close proximity to each other. And now this flow of blood is also in counter current pattern. The flow of blood in vasa recta and the flow of urine in the the flow of blood in the vasa recta and also the flow of urine in the nephron part is also in the counter current pattern, and this proximity also helps in the regulation of increasing or maintaining the increasing osmolarity between the medullary fluid and the urine. Now this is generally maintained by the ionic gradients. I have already told you in the reabsorption of NaCl and urea, those both are reabsorbed and secreted one after the other, like it is reabsorbed in some part and it is secreted in some part, in order to maintain this osmolarity only, in order to maintain the ionic gradients. The gradient here, the gradient is mainly caused by this osmolarity, increased in gradient is mainly caused by the NaCl and urea. NaCl is transported by the ascending limb of Henle, which is exchanged with the descending limb of vasa recta. See here, the ascending limb of loop of Henle is reabsorbing NaCl, and this reabsorbed NaCl is being transported or is being secreted into the blood in the vasa recta. Next part, in the vasa recta, the NaCl is returned to the interstitium by the ascending portion of vasa recta. Now when this NaCl, when this blood is flowing through the ascending limb, this NaCl is being again reabsorbed into the medullary fluid, and from here the same thing happens. See here, again it is reabsorbed, and this counter current pattern which is followed here, the NaCl is reabsorbed. When you only focus on vasa recta, the ascending limb is reabsorbing NaCl, and this NaCl of the ascending limb, I have got disturbed from in the live chart. Wait guys, Yes, Dr. Rohit Roy. I'm actually very poor in Hindi, man. So, yes, I have understood your message that you are saying you are actually from Hindi medium and you are not able to understand or you are not able to follow me, which I am speaking in English. So, I am very poor in Hindi. So, yes, what to do? Okay, this channel is basically started for English medium, so that is let us keep it aside and then continue. When you only take the vasa recta by putting the nephron part aside, in the ascending limb of vasa recta the NaCl is being reabsorbed, and in the descending limb of vasa recta the the same NaCl is being secreted. Now when you compare both vasa recta and nephron, NaCl is being reabsorbed by the ascending limb and it is secreted in the vasa recta. So these two proximity between the vasa recta and the counter current flow direction between the vasa recta ascending limb and descending limb and the ascending limb and descending limb of loop of Henle, this increasing osmolarity is maintained. This increasing osmolarity is maintained. The same thing is happening with the urea. The same thing is being happening with the urea. The urea is being reabsorbed by the collecting duct, and this same urea which is reabsorbed by the collecting duct, it is being secreted into the ascending limb. It is being secreted into the ascending limb, and this is how the counter current mechanism is being maintained in order to is being followed in order to maintain the increasing osmolarity towards the inner medullary interstitium. Okay, I hope it is very clear till now. If you have any doubt, please let me know in the chat box now. If you have any doubt, please let me know in the chat box. We'll be starting with the regulation of kidney function. This maturation, it is nothing but it is excretion of urine. You read this, guys. You read this maturation. I'll be completing now the regulation of kidney function. Now I'll be completing the regulation of kidney function, and you will be left with the role of other organs and disorders. These two I'll be completing in the recorded lecture, guys. In the live lecture, in this lecture I'll be completing, or else let us complete, guys. Come on, let us complete. I require a small encouragement from you guys. Come on, please encourage me. Encourage me, guys. Come on. So 1 minute, guys. 1 minute. I have some messages. We'll be completing this chapter now, guys. In the next lecture, we'll be starting with the as per our schedule. We'll be starting the living world. As per our schedule, we'll be starting with living world. And no one is responding, so I hope it is very clear to all of you. Now we'll be moving on to the regulation of kidney function. Regulation of kidney function by different types of hormones. So for this, this NCERT is okay. This is not that important. First now, let us move back to the bout. Original bout. Okay. Let me switch switch off this. Let me switch on this. Yeah, let us move on further. Yes guys, now we have completed with this. Are we clear with this? Yes sir. Are we clear with this? Yes sir. Okay. Okay. I'll be starting guys. Wait. Yes, now let us start. Now let us start with the regulation of sorry, kidney function. Regulation of kidney function. Basically, we will be discussing different types of hormones which affect the functioning of kidney by hormones. We will be discussing these guys. We will be discussing basically the hormones which affect the functioning of kidney. Okay. The hormones which we will be discussing in this part are ADH, antidiuretic hormone or vasopressin. Next one, renin. This renin is also called as RAS mechanism. ANF. The third one is ANF. These three hormones we will be discussing in this under this subsection. Under this subsection we will be discussing about the role and effect of these three hormones on the functioning of kidney. Now starting with renin, we will be then okay, let us start with ADH guys. Let us start with ADH and then we will be moving on to the renin. Okay, ADH. Actually, before starting this, how you can say how our brain knows that more amount of water is being secreted or more amount of water is being released through urine. How our body comes to know? This is because of the osmoregulators which are present on our body, and these osmoregulators get activated whenever large amounts of body fluids are being excreted out through the urine, and these send signals to the brain. And we have discussed the function of ADH, that ADH is antidiuretic hormone which is released by come on, tell me. ADH and ADH is released by, yes, by the hypothalamus. It is secreted by hypothalamus and it is stored in the posterior pituitary gland. Am I right? It is stored in the posterior pituitary gland. It is released by the posterior pituitary gland, but it is secreted by the hypothalamus. Now this is also called as vasopressin. Now, whenever these osmoregulators get activated, they send message to the brain in order to release ADH, and this ADH gets released from the brain. And when this ADH is released, this is an potent vasoconstrictor. ADH, 1 minute guys. This ADH is a potent vasoconstrictor. Guys, this ADH, it is a potent vasoconstrictor, and whenever the GFR falls, GFR, glomerular filtration rate falls, and the water is being more excreted through the urine, and these osmoregulators send message to the brain. ADH is secreted, and this ADH, being a potent vasoconstrictor, it can constrict the blood vessels and facilitate more absorption of water, and also it facilitates the increase in the GFR, and ADH also, it helps in reabsorption of water. It stimulates reabsorption of water. By these two mechanisms, ADH can increase the GFR. ADH can increase the GFR, and it helps in the loss of body fluids through urine. So this is your first hormonal mechanism, or which is affecting the function of the kidney. So now let us move on to the second one, about renin. When there is a fall in GFR, GFR falls. When there is a fall in GFR, the juxtaglomerular cells, these juxtaglomerular cells are the cells of juxtaglomerular apparatus. In this juxtaglomerular apparatus, they are the cells of, come on, please fast, they are the cells of the afferent or efferent arterioles. Now these juxtaglomerular cells secrete renin. These juxtaglomerular cells secrete renin, and this renin converts angiotensinogen to, this is an inactive form. Yes, anything which is ending with nogen, like pepsinogen, it is your inactive form of pepsin. This is your inactive form of angiotensin 1, AT1. Angiotensinogen to angiotensin 1, it is done by renin. And this AT1, it converts into angiotensin 2 using angiotensin converting enzyme. And this angiotensin 2, it is again a potent vasoconstrictor. Now, what are the two vasoconstrictors that we have came across? ADH and AT2. These two are the potent vasoconstrictors. These two are the potent vasoconstrictors. This is one mechanism through which constriction of blood vessels and increasing GFR. This is one mechanism. The second mechanism is it stimulates the adrenal cortex in order to stimulate aldosterone, and this aldosterone secretes Na plus into the blood, and due to more concentration of salt in the blood, the blood vessels contract, and therefore GFR again increases. And this mechanism of increasing the GFR through the angiotensin and aldosterone is called as RAS mechanism. This mechanism is called as RAS mechanism. R means renin, A means angiotensin. Next A means aldosterone, and S means system. Very important RAS mechanism. The pathway is very important. Like first renin comes, it converts angiotensin to angiotensin 1. Then comes the ACE, angiotensin converting enzyme, and this angiotensin converting enzyme converts angiotensin 1 to angiotensin 2, and this angiotensin secretes the aldosterone. This is your third A. This 1A, R means renin, A means angiotensin converting enzyme, another A means aldosterone, S means system, and A mechanism. This is your RAS mechanism, and this is how the renin affects the kidney function. And now moving on to the third hormone which I have told you, that is your ANF. Now this is your last hormone, and this would be your last hormone. Come on, the third one which we are studying is your ANF or ANP. This ANF or ANP, the full form is arterial natriuretic factor. Arterial natriuretic factor or peptide. This is generally secreted by the right atrium of the heart, and this is having an antagonistic function to the previous hormones which we have discussed, that is ADH and AT2. Now this ANF, it is a potent vasodilator, but those are vasoconstrictors, potent vasodilator, guys. This ANF is a potent vasodilator, and whenever the BP increases, whenever the blood pressure increases, GFR rate increases, whenever there is much of water in the body and it needs to be excreted out, so then this ANF is released, the BP is increased, and this BP, blood pressure, when blood pressure increases, in order to dilate the blood vessels, this ANF is released, and when this ANF is released, there is a decrease in the GFR. So that is coming to the regulation of kidney functions by hormones, guys. That is for this subtopic. Now we have the last two subtopics. One is your disorders and 1 minute, let me erase this. Disorders and the role of other organs, guys. And these two I am planning to do in the next lecture, guys. Role of other organs does not take much time. I'll be giving you the table, I'll be reading in, then you have to by heart the table. It is by hearting part. And disorders I'll be explaining you. Disorders would be remaining in the next lecture, that is your 8th part of the video. And yes, that is it for this live stream. Thank you one and all for joining my live stream, and if you like it, please, yes, there is only one like. If you like it, still, if anyone is watching the recorded thing, I'm telling to you, if you like the video, please hit that like button and also subscribe to our channel by a lab. And I'll be back with another lecture. That would be recorded lecture most probably, and then tomorrow morning I hope, I'm planning to upload it. Tomorrow evening we'll be starting with the new chapter, freshly new chapter, that is your living world. Okay, so that is it, guys. Thank you. Have a great day further. This is Vamsi signing off from Biolab, and have a great day further, guys. I'm ending the stream. If you have any doubts, please mention.